Thank you for watching. My name is Jenna Gallegos, and I am a postdoc in Jean Peccoud's lab at Colorado State University. Our lab is really interested in the intersect between cybersecurity and biosecurity. This is a recording of a recent talk I gave at the Synthetic Biology, Engineering, Evolution, and Design meeting about our attempts to apply digital signatures to DNA. First, consider whether you or someone in your lab has ever wasted time or reagents only to discover that the sample or strain you were working with isn't what you thought it was. Maybe there was an unexpected mutation, the tube was mislabeled, or the sequence was just poorly documented. These type of quality control issues happen all the time. We're attempting to address them using cryptographic techniques. Imagine this hypothetical scenario. A postdoc sees a plasmid described in a well-respected journal that she wants to use in her experiments. So she emails the corresponding author. Unfortunately, the graduate student who made the plasmid has long since left the lab, but the PI manages to dig up an aliquot of the plasmid and very graciously sends it. She uses the plasmid in a series of experiments. Several weeks go by, and her experiments don't seem to be going as expected, so she begins to doubt the integrity of the plasmid. What can she do to restore her confidence? The plasmid was likely documented in the paper one of four ways. Most likely, the relevant features of the plasmid were described. If she's lucky, there might have been a description of how the plasmid was constructed in the methods. But it's very unlikely that the paper included a map of the plasmid, and it almost certainly did not include the full plasmid sequence. For the sake of argument, we'll assume the manuscript included a plasmid map. So the postdoc does a series of restriction digests, and she finds that the plasmid matches the expected digest pattern. But she still can't confirm whether the features that are important for her experiments are unchanged. She could sequence the plasmid, but without the original sequence to compare it to and without annotations, she won't learn much. So, after several more weeks of failed experiments, she finally gives up on the plasmid. Most researchers are anecdotally familiar with a scenario like the one I just described. And there's a general consensus among life scientists that reproducibility is an issue. Some companies have even started offering authentication services to address these issues. But these efforts have focused mainly on confirming known variants in mammalian cell lines. Really confirming the integrity of an entire genome or a DNA sample requires access to a digital sequence file. Those files are often not shared with a physical sample, and even if they are, there's no guarantee that there wasn't some kind of mix-up. So the question becomes, how can we inextricably link a physical DNA sequence to its digital representation? Cybersecurity may offer a solution. In cybersecurity, digital signatures are used to enable authentication of a digital file or web page. Digital signatures allow the recipient of an email, for example, to confirm that the email came from who they thought it did. They also ensure that the message was not altered intentionally or unintentionally during transit. There's already a precedent for applying cryptographic methods to DNA. We're working on developing an approach to apply digital signatures to DNA in living organisms. Let me step back for a moment and explain digital signatures for those of you who may not be familiar. To generate a digital signature, a hash algorithm is applied to an electronic file like an email or a web page. The resulting hash value is combined with the signer's private key to generate an encrypted hash, and the encrypted hash is added to the file. For the validation process, the encrypted signature is extracted from the file. The hash algorithm is used to regenerate the same hash value from the file, and the signer's public key is used to decrypt the hash. If the resulting hash values match, the signature is valid. To test the feasibility of applying digital signatures to DNA, we've developed a pilot software that generates digital signatures for plasmids. In this scheme, the signer provides several user inputs, starting with their ORCID. ORCID stands for Open Research and Contributor ID, and ORCID is a nonprofit organization that uniquely identifies researchers using a 16-digit number. Most of you are probably familiar with your own ORCID, since some funding agencies and academic publishers are beginning to require them. The plasmid ID is a 6-digit number that serves as a unique identifier for the plasmid. This number might be associated with the plasmid in a laboratory information system or a plasmid database like AdGene. This is the ID that will be used to describe the plasmid in publications. In the pilot version of the software, the user also supplies start and end sequence delimiters. These sequences will help the validation software identify the signature down the line. We're working on developing a system where the start and end delimiters will be fixed instead of user selected, which will simplify the validation process. The user then specifies the location within the plasmid where the signature should be inserted in order to ensure it doesn't interrupt any important features. Finally, the user specifies how many errors they want to detect. Included within the signature is an error correction code. This relies on algorithms like those that are used to encode redundancy within CDs or DVDs. In this case, however, the error correction code will merely be detecting errors, not correcting them. Assuming a minimal number of mutations occur between signing and validating, the error correction code will return information about what and where those mutations are. I'll show you what that looks like in a minute. The user specifies the number of errors they want to tolerate because the length of the error correction code increases depending on the number of errors that can be detected. Once the signature is generated, the output looks something like this. The start and end delimiters flank the insert, and they match the sequences specified on the previous slide. The ORC ID is encoded by 32 bases, and the plasmid ID is encoded by 12, corresponding to 2 base pairs per digit. The error correction code is relatively short, just 32 base pairs in this case. And the signature is always 512 base pairs regardless of the size of the sequence being signed. That brings the total length of the signature to 588 base pairs. The signer can then order the signature from a DNA synthesis company and insert it into the plasmid. Let's return for a moment to the hypothetical scenario I introduced at the beginning of the talk and discuss how it might be improved with the use of digital signatures. If the graduate student who made the plasmid was conscientious enough to sign it, the postdoc receiving the plasmid could have sequenced it and uploaded the FASTA file. The software would then search for the presence of a signature. If there's a signature present, the identity of the signer and the plasmid are extracted. If the sequence that was uploaded matches the original sequence of the signed plasmid exactly, the signature is valid. If the sequence is changed in some way, the IDs are still extracted, but the signature is invalid. If the number of errors is lower than that set by the error detection threshold, then the user gets a pop-up box that returns information about what errors occurred and where they're located within the sequence. If instead there are more errors than that set by the threshold, there is instead a message that simply says there are too many errors. There are a number of outstanding questions at this point. First, there are several ways that inserting 588 base pairs of extra sequences might impact the plasmid. These include retention of the plasmid, the function of the plasmid in terms of what protein products are produced and how much, or the stability of the signature or the plasmid itself as it relates to propensity for homologous recombination or mutations. Additionally, we've so far only focused on plasmids. Applying digital signatures to microbial or even eukaryotic genomes introduces a number of additional challenges. For one, the mere size of a whole genome increases the likelihood of mutation, and sequencing technologies are not yet robust enough to provide the kind of single nucleotide level validation that digital signatures would require for an entire genome. However, you can imagine how this technology might be applied to just particular portions of a microbial genome, such as those that have been re-engineered for a specific purpose. We've designed a series of experiments to conduct initial validation tests. We'll be inserting a signature into the garden variety vector PUC19 and designing a minimal expression vector which includes a signature. We're also ordering a family of signed plasmids with various mutations in different locations. These experiments will allow us to confirm that a mutation anywhere invalidates the signature, and also that the error correction code can detect an error regardless of its location. If anyone has additional ideas for experiments they'd like to see, we'd be happy to discuss those with you. I want to close in emphasizing that this is just a proof of concept. We've been considering a lot of possible applications and deployment strategies for this technology, and if anyone has ideas about how the technology might be used or expanded or improved, we'd love to hear them. Additionally, this project is just a small part of a larger initiative to address issues at the intersect of cybersecurity and biosecurity, which we discussed in a recent paper in Trends in Biotechnology. We're working to put together a Center for Cyber Biosecurity at CSU, and we'd be happy to discuss that further as well. Funding for this project, as well as the Center for Cyber Biosecurity, is coming from a Catalyst program within the OVPR at CSU. I'd like to acknowledge my PI, Jean Peccoud, and we're very grateful to our collaborators in the Computer Science Department. Diptendu Kaur is the master's student who has developed this pilot software, and his advisor, Indrajit Ray. Thank you for listening, and if you have any questions, I'd be happy to take them in the comments section, or feel free to contact Jean or myself directly. Thank you.